This is TWIS. This Week in Science, episode number 877, recorded on Wednesday, May 25th, 2022. Don't forget your towel or science. Hey everyone, I'm Dr. Kiki, and tonight on TWIS, we will fill your heads with termite trees, bird buds, and pea pals. But first, thanks to our amazing Patreon sponsors for their generous support of TWIS. You can become a part of the Patreon community at patreon.com/thisweekinscience.
Disclaimer, disclaimer, disclaimer. If you've been hitchhiking through the galaxy, you'll know that preparation is key. Expect the unexpected and you'll never be caught off guard. Know there are unknowns and you'll always be knowledgeable, or you'll at least feel better about facing the probable crapshoot that is your future. Look back for inspiration, but don't get dragged downhill. Look forward with optimism and be ready for change. Look outward, look inward, look upward, look downward, look skyward. The last one will keep things from landing on your head and just might help you catch your next ride. But only if you don't forget your towel. And the latest episode of This Week in Science, coming up next.
I've got the kind of mind I can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening. What's happening? What's happening this week in science? What's happening? What's happening? What's happening this week in science?
Good science to you, Kiki.
And a good science to you too, Blair, and everyone out there. Welcome to another episode of This Week in Science. We are back again, minus a Justin, as he is soon to be in transit back to the United States. But we are moving forward with our science and our towels. So let us move into the science for the week. I have stories about AI moving science forward, termite travels, bird stuff, and more. I've got a lot more, but we'll get to it. What did you bring for the Animal Corner, Blair?
Oh, I for the Animal Corner, I brought dolphin pee and baby turtles. But I also have a fun story about skydiving salamanders and also the hygiene hypothesis, return of the hygiene hypothesis.
We've never talked about that before.
Oh, yeah.
Once or twice. Okay, I'm looking forward to it. We're going to clean up with our science.
Not too much.
Not too much, just enough. We're just enough cleaning. That's right. As we jump in, I want to let you all know that if you are not yet subscribed to This Week in Science, you can find us all places podcasts are found. Look for This Week in Science. You'll also find us on YouTube, Facebook, and Twitch, where we stream weekly on Wednesdays at 8 p.m. Pacific time. You can find us on Twitch, Instagram, and Twitter as TWIS Science. And if all this is just so much to fill your head with, then just go to our website, which is twis.org. All right, you ready to jump into the science?
Yes, give me the short stories.
The short stories. All right, let's start with some artificial intelligence. Just really moving science forward and potentially leading us to, the future of science, really. I mean, that's the bigger picture. But the short stories that published this week in Nature Astronomy is a paper by Joshua Bloom and his colleagues showing how a machine learning inference algorithm, an artificial intelligence algorithm, was able to find details related to gravitational lensing that scientists over the decades since it was first shown by Albert Einstein have not picked up on. So gravitational lensing is the way that we are able to use the gravitational field around a star or an object and how it bends light into a new trajectory as light passes around that object. Light is affected by gravitational fields. Fascinating, right? So the idea with searching for exoplanets is that we can use one star in front of a planet that passes in front of one star. And as that happens, we're able to see the dimming of the light. When there is a star in front of a star with a planet going around, then you have a gravitational influence that occurs because of the star and the orbital mechanics of the planet around the star that it's orbiting. Anyway, astrophysicists for decades have basically said, okay, there's a couple of outcomes to this, and they split it up into a couple of different categories. The AI said, it's more complicated than that. Which
Silly humans.
Silly humans. You're missing out on all sorts of nuance. And so now, it doesn't make things easier for astrophysicists who are trying to figure out the exact location and orbital mechanics of transiting planets in front of stars, transiting other stars. It doesn't make it easier on them. It makes it a little bit more complicated, but it's going to be more accurate over time. So the way that normally the orbital stuff is worked out with this gravitational, in this sense, gravitational microlensing, they call it degeneracy. And so instead of just two degenerates, there's just a whole posse of degenerates that are now leading to a whole unifying theory for this gravitational lensing instance.
AI showing up the humans.
Yeah. And so really, as I alluded to right in the beginning of this story, is that there are many things where humans, we think our pattern recognition is fantastic, but when you start looking at massive data sets and when you start looking at the various combinations, when there are lots of parameters involved that make it more complicated, that's when the machine learning can really start discovering things that are outside our abilities. And that's what's going to, humans in conjunction with artificial intelligences are going to push science forward. Mm-hmm. Are you afraid?
Always.
Don't fear the AI.
If they figure out the physics before we do, then we're really doomed.
Why?
That's, they'll figure out theoretical physics, yeah. And they'll just start doing things that looks like magic to us, but they're understanding the universe in ways we never have. We won't stand a chance.
Well, if the artificial intelligences have already figured out time travel, wouldn't we know it?
Unless they don't want us to know it.
Mm.
Maybe it's all playing off as it is supposed to. Yeah. Anyway.
All right. So you don't want to live with an AI, but what about you live with a dog?
Oh, I love it. And it turns out it's good for you because living with dogs or large families is gross. And that's a good thing.
What? Why is it dogs or large families?
I'm going to tell you.
Okay.
So this is a study looking at the hygiene hypothesis, something we've talked about a lot. Mm-hmm. The idea is that lack of exposure too clean of an environment growing up, lack of exposure to microbes early in life actually leads to immune issues. Because if you're not exposed to those things growing up, then your body overreacts when they show up later on, essentially. And so researchers used an environmental questionnaire to collect information from nearly 4,300 relatives of people with Crohn's disease and enrolled in the Crohn's and Colitis Canada Genetic, Environmental, and Microbial Project, the CCC-GEM project. Okay. They specifically wanted to look at Crohn's disease because it is an inflammatory disease in the gut that they think could have something to do with this. They analyzed several environmental factors, including family size, presence of dogs, presence of cats, other household pets, numbers of bathrooms in the house, whether they lived on a farm, whether they drank unpasteurized milk, whether they drank well water. And they also looked at the age at the time of exposure for all of these things. What they found, again, this is purely a correlation from a self-reported survey. So this is a lot of asterisks. This is not direct evidence. But what they found from this survey, this questionnaire, is that exposure to dogs, particularly between age 5 and 15, was linked to healthy gut permeability and microbe balance, and the body's immune response as well. All those things are protectors against Crohn's disease. They did not see that result with cats. They're not sure why. But
that's interesting. Yeah. I wonder if it's the different proteins in the saliva that usually become allergens. I don't know. This is interesting.
So it could be that.
Okay.
They suggested it might have something to do with the activities that owners generally get up to with dogs versus cats, and that dogs force owners outside. So you get more environmental exposure. I also think it's because people roll around with dogs, and dogs are pretty gross. And dogs will like lick you on the face all the time, right after they've licked their butt. Like, even if you want to believe my dog doesn't do that, it's happened. Okay, it's happened. Get over it. But that's my theory. But it is also true with families of 3 or more members in the first year of life. I would argue similar, just grossness, germs, picking your nose and then touching your sibling's face, just like all of that, right?
Kids not washing hands, lots of just, the twin rivers, the 11s streaming down the nose, like all the things.
Yes, exactly. But so this is what they saw, dogs and larger families. They hope that these findings will help us physicians kind of know what questions to ask when they are determining who is at high risk for gut issues. So if you say you have a dog, that actually could be relevant medical information. Okay, you have a dog, you are less likely to have Crohn's disease. This might impact how I treat your symptoms, or it might impact how we look out for it as you age, right? So it's, even if you can't figure out the exact kind of mechanism right away, it could still help with medical care if you know there is a correlation. You can kind of test it out, figure out if that correlation might be causative, and then you can use that information to impact your care. So it's still way down the line. This is really just, hey, when do you think you got a dog? And how old is your sibling?
At what age? At what age were you first licked on the face by an animal?
And
what kind of animal?
When did you drink well water?
Right.
So it's definitely subjective still, but it's a very interesting addition to the hygiene hypothesis where we talked about dishwashers before and all that kind of stuff. Just kind of feeds into the general theory that it's good to be exposed to some germs when you are growing up, and I like irritants.
I like your interpretation that, it's whether or not you're growing up in a gross situation. Yeah. Dogs and family. Rah.
Yeah, it's something I've actually thought a lot about with COVID too, because if kids spent over a year inside, and so that was a long time that if they were a single child home without a dog, they were not getting exposed to very much irritants potentially, as opposed to if they had other siblings at home, they could have been exposed to more, or if they had a dog or 7, I don't know. So it's a whole, there's an impact there because there also could be getting exposure at school, of course. So when you take that away, it changes things.
You take it all away, reduce the exposure. Suddenly, we'll see. We'll see what happens with immune systems and allergies and autoimmune and gut disorders as the generations kind of pass. What's going to happen 40, 50 years from now?
Yeah.
Right?
Yeah, you have to get your inoculation when you're 5 of germs, basically. There's a cocktail we mixed up of all the potential allergens in the region that you live. Yeah, exactly. Think about that. You could get exposed to allergens. You could get exposed to low levels, basically just a vaccine, right? Low levels of potential diseases, and then also any gut irritants or anything like that. Just expose it all when you're young.
Yeah. Well, speaking of digesting things and how that works, let's talk about termites. Wood termites.
Oh, yes. The scourge of many a homeowner.
Yeah, homeowners tend to not like them very much.
No.
However, some researchers at the Evolutionary Genomics Unit at the Okinawa Institute of Science and Technology Graduate University, with collaborators from around the world, find wood termites very interesting. And they have published their research looking into the phylogenetic tree, the evolution of wood termites in molecular biology and evolution. Dry wood termites, they are the second most specied termite species. And Blair, did you know that termites come from cockroaches?
No, but they look similar. They have a similar body plan, right?
Yeah, so you say they look similar, and that's really a lot of the information that we have about termites and their evolutionary history. It has to do with morphology and what they look like and where we find
So
them.
we could be totally wrong.
Right. So anyway, termites are a type of cockroach. They split from cockroaches about 150 million years ago, which I find fascinating. Did not know that. That's new, just trivia point that people out there, you just take that one, stick it in the brain bank there. These drywood termites are known to form smaller colonies, usually less than 5,000 individuals, as opposed to other termites, which live in the soil in big tunnel colonies, and they're all in the underground. And those can be like thousands and thousands and thousands of individuals, millions of individuals. So drywood termites tend to be, not quite as social, I guess, but they're still pretty social. They're otherwise known as the Calotermitidae. And the Calotermitidae, because they split fairly early, so it was like the split from the cockroaches, and then around 100 million years ago, they split off from the other termites. And it was this very early split. And so they were thought to be really primitive without a lot of interesting behaviors or other things aside from like eating wood and digesting it and reproducing. That's about it. Anyway, the researchers were like, hey, let's check this out. And so they collected about 120 different species, multiple samples from around the world, and were able to really look at the diversity of the various species and figure out what started where and when and how. And it turns out that they have been all over the world. They didn't just start in different places. They started in South America. These termites first split off. Drywood termites first split off in South America, and then started moving around. And because they live in wood likes to float. They have been across the oceans of the world some 40 times in the last 50 million years. So they've floated back and forth on bits and chunks of wood. And then more recently, with our wood-holed boats and our lumber that we take from place to place, humans have helped them spread from place to place around the world. And so they actually have a very interesting genetic history. A lot of the diversity comes from these various, I guess, ocean travels, starting in one place, going someplace there, then dividing out into multiple species. Oh, and then coming back and interacting maybe with an older species. And suddenly you have these species that are sub-lineages of each other kind of getting along. And then you have cross-pollination. And then so their diversity is fascinating. And it turns out that they're not that simple. They actually have a lot of the complex behaviors that were not expected to be had in these multiple species. So, hey, who knew? Drywood termites, they're diverse, they're fancy, and they like to travel.
They have a braided stream way better than ours, it turns out.
They do.
We get all excited. We're like, well, it's not just out of Africa. We also bounced around in these areas and over here. It's like, termites have been around the world 50 times.
They have. They've been, and they've really been all over the world. I mean, for a species that started in South America, they've traveled around the globe. They are everywhere. And
not in Antarctica, though.
Right. Except for Antarctica. There's not much wood for them to chew on down in Antarctica. So that wouldn't
There's just
make
that
a very
one
good
ship we
vacation.
talked about a couple weeks ago. Just
Yeah.
that one. It might pop up.
It might. Yeah. So the interesting question now is we've got a little bit more understanding of how interesting their history is. So now the textbooks that include termites need to be rewritten because their phylogenetic history, which was based on morphology or how they look, is wrong in a lot of ways. So time to rewrite some of those termite textbooks made from trees, which termites like to eat. Oh, it's the circle.
So you really do need the latest entomology textbooks.
You do?
Yeah. Yeah. It's some of those other things. You don't need the latest English lit textbook. Last year's was fine. History, maybe that last chapter is important, all that new stuff. Biology, science, it can change drastically from year to year. So you got to get that new
Here we
book.
go. One study, suddenly there is so much more understood about the termites. So termites are one thing. Salamanders. They're another. I love salamanders. Tell me about them.
Yeah. You know what you might not have understood about salamanders before? They fly now.
What?
Yes. So
like
the
snakes.
wandering salamander, what a perfect name for it. They hang out at the top of redwood trees. And a new study from the University of South Florida has found that this highly arboreal, that means living in trees, species of salamanders, specifically the wandering salamander, Anades vagrans, engages in parachuting and gliding to slow and direct their descent from the trees. So climbing down, that's for suckers.
Who does
I'm
that?
a jump.
I'm just going to jump. It's a lot. It's a lot of work. Let's go.
Yeah. So they rely on postures like skydiving humans to slow and control their fall. This really hasn't been looked at salamanders ever. For one very simple reason, their body does not look like it lends itself to parachuting or falling vertically without dying at all. They are little water monsters. That's their deal. They are perfectly adapted for swimming. They have that blade-like tail that helps them to swim through the water. They can kind of scurry on land, but they have that sideways joint of their arms and legs, so they're not even, that effective at that. So all of this to say, why would you check to see if they're good at falling? Well, it was observed in the wild, and so it was time, you guessed it, to put salamanders in a wind tunnel. Yes.
Of course.
Of course. They parachuted consistently, slowing their vertical speed by up to 10% while they fell. They coupled parachuting with undulations of their tail and torso, which allowed them to affect their gliding at non-vertical angles at about half of the time. Not only could they slow themselves down, but they used fine scale control in pitch, roll, and yaw. That's basically just all the directions when you are falling to maintain upright body postures. Yeah, exactly. It's just 3D is all that means.
X, Y, and Z axes.
They also executed banking turns, and they were able to glide horizontally. So this is beyond just not dying. This is being very in control of their orientation and placement in the air. And again, this is the first time that this has been observed in salamanders. And now I would say the thing that's really cool about this is that it's time to reevaluate any animal's ability to do this, because if you see this video, it does not look like an animal that is well adapted for this. You would not expect it by looking at a salamander that they would be good at parachuting, basically skydiving without a parachute, excuse me. And so that means almost any animal could potentially be capable of this because they don't have any fleshy wing-like or webbing-like or fin-like appendages that should help them with this. They are really just using their movement to slow them down and to manipulate themselves in the air. So in theory, anybody could figure this out.
I'm wondering, though, I mean, they live in the treetops. You don't want to just die if you fall.
Right.
So
I mean,
What
so the
other
ones
animals?
that don't
I know
die
there
are immediately selected for
birds
in the
fly,
strongest
right.
way possible.
Right. But if you're lightweight, so we know that insects very often can survive falls from great heights because of their mass to, body size, the ratio that's there. Are the salamanders the same way? This particular salamander, could we take salamanders, tiger salamanders that normally stay on the ground?
They're so chunky. I don't know if they'd be able to do it.
Yeah. So is this just a body size and they have a reflex where they spread out like a skydiver? Is that something that makes it more possible, whereas other salamander species would just plummet?
Right. Well, because that's the other problem, right? Is that we humans can't skydive without a parachute because, of the whole momentum thing. So we're so heavy and dense that as we fall, there's an acceleration and that creates a larger force over time. And so there's that whole issue.
Mass times acceleration squared.
Yes, exactly. There you go.
Mass times gravity. Yes. Anyway, yes.
And but yeah, so I have a similar theory that even if the tiger salamanders spread out perfectly out of instinct, it still might have a bad time because it is so much chunkier. These guys are small. They're like as big as your finger and they're very thin. So I do think part of it is just, a feather does fall slower than a bowling ball when you are not in a vacuum.
Not in a vacuum, no, but with air resistance, that makes all the difference. And that's what we're talking about here is, natural atmospheric conditions.
Yeah.
Fascinating. Parachuting. They've got they're little salamanders with squirrel suits. No.
Yes. Yeah. So next time you go into a redwood forest, look up or don't, because there might be falling salamanders. For me,
look skyward. You'll be looking up
to me. For others, they might not appreciate getting bonked on the head by a salamander. I would love it. It would make my day.
I would love to catch one on my head for sure. For sure. All right, Blair. You might not plummet to the earth like a salamander when it's your time to go, but we all have aging ahead of us. That's just part of life. Even though some people want to avoid aging, it does happen. And so researchers are trying to figure out, what's going on inside the cells with respect to aging and how it happens. Well, we know that there is, our chromosomes are packed tightly into these structures called chromatin. And so the chromatin is all tightly wound around itself and around and about, and all of the molecular interactions allow it to take this really tightly packed shape. Some of it is epigenetics that wrap around structures called histones. We have glycosylated sugared areas that stick out, and all of these epigenetic areas are part of kind of what makes us who we are, makes our bodies work the way that they do. And epigenetic stuff, while we can inherit some of it, a lot of it is stuff that might come about as we develop and as we grow. So the aging epigenome is something that's of a lot of interest. And these researchers just publishing in Developmental Cell, we're looking at large-scale chromatin reorganization around the aging epigenome. And they found that with aging, there's hierarchies in the chromatin. So some areas are more preserved, or their packing and their structure is preserved better and longer. But as you age more, there's more entropy and epigenetic instability as you age. So things that were wrapped around histones, maybe they're like, boop, I don't need that histone anymore. And so suddenly you have areas of your genes that are able to be turned into proteins that didn't used to be. And that can be bad. Or it contributes to aging, really. Along this, they found that as the deformation happens, there is basically this entropy, or what used to be structured areas within the chromatin. It just gets lazy. And so the structure starts to go away. And additionally, they discovered there is a placenta-specific protein type that starts to be expressed in your cells. Normally, this protein is only expressed within the placenta. And as you're growing in the womb, you, baby, are producing your placenta. And so when it's time for your placenta to stop growing, there are command signals that say, hey, placenta, stop doing the growth thing. We're good. And those are these same placenta-specific signals that start to pop up in aging cells with this old entropic chromatin. So it's like a circle of life thing that's going on. And I find it super fascinating that you're born and you have these control signals in your placenta that say, okay, no more placenta. It's time to be a baby and enter the world and do all this stuff. And then when it's time to start exiting the world, your cells are like, okay, let's do that placenta signal again. And it's time for us to, maybe exit the world.
No. That only works if you can hook me back up. 3D print me a placenta, please.
Get me a placenta. So
put it in a fanny pack, carry it around with me.
Yeah, I don't know. That'll be your fanny pack full of Anyway.
Yeah. No, you got it. I'm all about it. I'll take it.
I'll take it. Yeah, but it's interesting that the epigenetic degradation, this as the epigenome starts to unloosen and unwind, things that maybe were packed away related to the placenta by epigenetics markers over your lifetime suddenly are opened back up again. And that they're thinking, as a result of this study, that they can start looking for this, what they call pregnancy-specific beta-1 glycoprotein, PSG genes, as a biomarker for aging. So that if we're able to see how much of this pregnancy-specific glycoprotein you have, it could really let people know how they are aging, at what point in their life. And so we might be able to identify aging drivers and intervention targets as well. So it's not all over. It's, targets are good, right? We like the targets for the promises of Blair's forever youth.
So let me ask one clarifying question. So if you were able to reverse this signal, that wouldn't actually do anything to your aging. This is just a byproduct of your aging, right? This is a signal of it,
right?
Not a cause.
Well, it's also a cause because the action of this particular protein that is to stop the growth and the development of the placenta. And so it starts to turn off things that maintain your cells and keep the cells active and healthy, and instead allows things that degrade cells and degrade metabolism to start coming into play.
So this could lead to treatment.
It could.
Information. That's very cool. I like it.
Yeah, so it's one piece of the puzzle. It's not the whole puzzle. It's not stopping this chromatin degradation from happening in the first place, but it is a downstream thing. So part of the puzzle.
Neat.
All right, Blair, do you have another story here? Or did
I think it's up to you now.
One more. Well, I think it's, as we're talking about this, if you and I start talking more and more about what the next thing we're going to do is, eventually we'll come to a decision, right? Like a democratic decision, really, right? And if we had a larger group of people, we could all put our voices in. Everybody could start talking about what we wanted to do and maybe come to some kind of consensus and then do it and coordinate our actions.
Yeah. Yeah, there's usually a ringleader, but you can get a consensus out of people.
You can get a consensus out of people. Well, you can also get a consensus out of jackdaws. Jackdaws are a kind of corvid related to crows and ravens, and they are also, as a lot of cities may have crow springs and crow falls where the crows come through, and in the morning or in the evening you hear the calls of the crows as they all come to roost in the trees, or they all get up and they leave and they go to wherever it is that they're going. Jackdaws have this behavior as well, and so some researchers decided that they needed to look at what happens with the sounds that the communicative sounds that the jackdaws are making and the behaviors that they're showing. Does the amount that they caw, 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 talk to their jackdaw neighbors, does that make a difference in what they actually do? And it turns out, what do you think, Blair?
Yeah. They're
Yes,
social animals.
They are very social animals. And so in the mornings, when they are going to, they roost in the trees in the winter. And before they depart, there will be one or two birds that start the calling. And they call more and more and more. And as a larger and larger number of birds enter into the cacophony of jackdaw morning music, it does correlate to the probability that they're all going to get up and fly away and go away. So jackdaws, they yell at each other, "Hey, Joe,
time
you ready to go? Are you, Blair? You ready? Are we ready to go now?"
No, not yet.
Yeah, come on, Blair. We gotta go. Come on.
All right, I'm coming.
Exactly. You imagine a whole neighborhood got to get on the bus. How do you get everybody on the bus? Everybody yells at each other to get on the bus. Then they get on the bus. Jackdaws.
Yeah.
It's one of the few examples that we have scientifically examined of this kind of coordinated communication for mass behavioral activity, which I think it's fascinating, and that it seems as though we should be looking into this more and more, since there are so many species that do have birds, especially in their migrations, these mass movements on a seasonal basis.
Yeah. I mean, point a finger at any Corvid, something special is going on, especially socially, so right? Much. So just pour all the research into all the Corvids. I want to know. I want to know what they're doing. I want to know why they're doing it. I want to know how they're doing it.
Yeah.
Thank you, Corvids.
Thank you, Corvids, for telling us. And I do have another Corvid story about their tiny, clever brains for later in the show.
Yay.
Yeah. More bird stuff, bird brain stuff later in the show. This is This Week in Science. Thank you so much for joining us for this episode. We hope you are enjoying the show. If you are enjoying the show, please tell a friend. Bring a friend to listen with you this next week. We'd love to hear from them. Hey, Blair, you want some COVID news? You got your towel? Are you prepared for COVID stuff?
No COVID news. No. I guess at least it's not monkeypox news. Stick with the COVID.
Yeah, we're going to stick to COVID. I don't have a monkeypox section for the rundown yet.
Thank you, no. This towel's going to turn into a security blanket.
That's what I'm watching you. You're taking it as a security towel.
COVID, go away.
We would like it all to go away. Yes. Oh, wouldn't that be so nice? Well, few studies this week to talk about. First one on the docket is published this week in Cell Metabolism. Researchers were looking at what exactly is going on in diabetic individuals to lead to them developing more severe COVID-19 symptoms when they do get infected. And apparently what's happening is that ACE2 is upregulated in hyperglycemia in kidney organoids and patient renal cells. And so they assume, based on this study, that this is likely what's going on in vivo as well. So in people where you have hyperglycemia, you've got the ACE2 receptor, which is important to SARS-CoV-2 infection. Actually, they're getting more of them in the kidneys, more ACE2 in the kidneys. ACE2 is also related to water movement and metabolism within the kidneys. So it has these dual purposes that are very important metabolically, but also can be a problem in this particular case. And so with this particular study, they saw that there were increased changes to the metabolism as well within these kidney cells and in the kidney organoids that they created. And the ACE2 receptors that were upregulated in their lab experiments boosted cellular infection of SARS-CoV-2. And so it's very likely that this increased amount of ACE2 is leading to increased infection with SARS-CoV-2, specifically getting in through the bloodstream, through the kidneys, and in through those cells. And that the possibility, however, and here's the upside, is that now that they're seeing that there are these metabolism related effects that increase the ACE2 receptors, potentially there's a way that we can target energy metabolism within renal cells in patients to be able to decrease the likelihood of infection. So, or at least decrease or at least help treatment if somebody does get infected. So understanding more about why diabetes is in particular a problem with this virus is really important for being able to treat it more later.
Right, because as much as we would like it to just go away, it's likely going to be around forever now. So we have to find a way to protect susceptible communities moving forward.
Exactly.
Yeah.
Yep. Forever, ever. Well, even though it's going to be around for a lot longer and we don't really know what's going on with BA4, BA5, all these other variants.
BA17, no big deal.
There is research out currently, just came out this last week, that out of University of Wisconsin-Madison, published in Nature, that the BA2 subvariant is similar to BA1 in the severity and in its ability to cause infection. So we weren't really sure whether BA2 was the same as the original Omicron strain, and it pretty much is. So Omicron, less severe than Delta, more infectious. BA2 is the same way. So for most people who are probably seeing their neighborhoods, their communities with BA2, which is the dominant subvariant around the world almost. I mean, seven dozen countries, it's the dominant variant. That, as you're watching it go through, you might also, people are getting it more often, but not as many people potentially are ending up in the hospital. But hospitals are starting to fill up again, folks. So, maybe those masks can help, because we know that vaccines should not be the only protection that we have. So ventilation, masks, vaccine is great. And finally, we've talked about long COVID and risk for long COVID before, and there are various studies as to the percent of individuals who end up with long COVID. Some studies have said up to 30% of people who are infected with any type of COVID end up with long COVID. Other studies say as little as 7% of individuals who are infected end up with long COVID. Well, the question has also been raised as to how does vaccination impact these probabilities? And it's not as good as we hoped. So vaccination, according to a study out this last week, only reduces the probability of long COVID by about 15%. So it's a little, but it's not as much as we were hoping it would be. So vaccination, as I said, should not be our only line of defense. It helps. It's part of the whole system. But as cases are rising, masking, social distancing, ventilation, do what we can, everyone.
So also, the math here is important to pay attention to. If it's only 7% of people who get COVID have long COVID, and
it's more likely to be the people who are in the hospitals more often who have the long COVID. Yeah.
Right. And then it's 15% of 7%. that's still, it's a very small number still. So that is, it's relatively, it's a small number. But the other thing I was thinking about is if the vaccine is less likely to get you, if taking the vaccine prevents you from dying, it could actually push those numbers higher than it would be otherwise, because you have kind of this confounding variable now of you didn't die. So if you could magically figure out if people who died from COVID were going to get long COVID, you might find out that the difference is actually much bigger.
Yeah.
But because all those people die who didn't get vaccinated, they're not part of the factoring in of who gets long COVID and who doesn't, right? So it messes with the numbers a little bit so that you have this bigger physical number. You have this larger N as opposed to a percentage, right? Of people with long COVID who are vaccinated, because there's this whole group, this whole number of people who are dead. It hurts your brain to think about, but it totally influences the numbers.
Yeah. And these are the kinds of things that the epidemiologists, the public health statisticians, these are the kinds of things that they're having to take into account to try and figure all this out. Yeah. I will let their brain hurt. Their brain hurts. Their brains hurt for me.
Yes. And I will also say, long COVID still sucks. And I'm not trying to say that it doesn't. But if I was picking, I'd rather have long COVID than die. So it's
one over the. It's which
and it's tough because there's some long COVID
varying
that are
levels,
truly horrendous. But it still is a question of like. I think overall, it seems like the vaccine did an excellent job at preventing death, which was its main job.
Yep. And preventing death and reducing severe disease as well. So fewer people end up in the hospital. Yep, it is true. Oh, and monkeypox, by the way, yes, it's spreading, but it doesn't spread at all the same way that SARS-CoV-2 does.
Don't touch any strangers with rashes or anyone with a rash who's been out of the country recently. Just don't touch them.
And people who have been inoculated for smallpox are probably less likely to be infected with monkeypox, and the monkeypox is likely spreading more easily because we have a whole lot of few generations now that have never experienced smallpox because we eradicated it. We got rid of it, right? So now stores of smallpox and monkeypox vaccine are being brought out of our storehouses to enable vaccination, inoculation for various populations that are highly at risk. But if it's an obvious rash, pustules, those kinds of things, just stay home and call your doctor. Stay away from other people. People don't go rubbing up on each other at a rave. I don't know if that's how it is. This could be a myth. And I don't
sound advice regardless.
It's sweaty. Okay. Enough about that. This is This Week in Science. It's me and Blair tonight talking about all the science that we wanted to bring for Ladies' Night with our towels and science. Woo! If you're enjoying the show, please head over to our website, TWIS.org, and click on the Patreon link. Patreon is how listeners of the show, people who enjoy the show like you, help support what we're doing here. Patreon allows us to be able to keep track of you and to send you rewards, small ones, $10 and more a month, and we will thank you by name at the end of the show. There are other things that come in the mail, little fun gifties for various levels. You can check out how the levels of support work with you and your budget. But any amount really helps us to do this show every single week and keep it going. We really appreciate your support and can't do this without you. Thank you. All right, let's come back now. There's what is it? This time in the show where we don't want to talk about COVID. We don't want to talk about the monkeypox. We only want to talk about the animals. Yes. It's time for Blair's Animal Corner. with Blair.
She loves our creatures, great and small. Biped, millipede, no pet at all. If you want to hear about animals, she's your girl. Except for giant pandas and squirrels. There's that up
What you got, Blair?
Oh my goodness. Would you like to hear about dolphins as pea pals? I knew you would.
Yes.
Yes. This is all about bottlenose dolphins. Always fun stories from our buddies, the bottlenose dolphins. And a recent study finds out, this is from University of St. Andrews, that they taste the urine of their friends in the water to figure out who's around. They keep their mouths open and sample urine for longer from familiar individuals than unfamiliar ones. And so this is the first case, as far as these researchers know, of a vertebrate shown to have social recognition through taste alone. Now, this is where, Kiki, it's time to put on your skeptic hat, because here is their reasoning. Dolphins do not have olfactory bulbs.
Wait, what?
Leaving the team certain it was taste and not smell at play. Now, okay. Let's
They're
back
under
up.
the water, blah, blah, blah.
Okay.
But
They don't have olfactory bulbs. First of all, we've played that game before.
yeah, we have.
With birds. So I don't know. We'll see. We'll see
Okay.
if truly they do not have olfactory bulbs. But even that, the thing is, this sounded very similar to me to things that other mammals do. And I'll get to that in a second. But just to kind of explain what these dolphins are doing, they, the bottlenose dolphins, they are, so first of all, this was a good individual species to look at this in, because they already know that they have signature whistles for specific individuals. So they remember individuals, which is hard to test in other species in general. How do you know this dog from that dog, or this cow from that cow? And so because dolphins have been proven to have these whistles that are almost like names for individuals, right? And that they can remember them for over 20 years, this means they have this baseline understanding of individuals and can tell individuals apart. So then they wanted to see if they could tell them apart by their urine. So they presented 8 dolphins with different urine samples from familiar and unfamiliar individuals. They found that they spent about 3 times as long sampling the urine from those that they knew. They're really swishing it around to try to figure out what's going on.
Oh my goodness.
So the basis for this, that they might care about the urine, also comes from something that's been observed that's called genital inspection. So this is when dolphins use their jaw to touch the genitals of another individual. And in many situations, there is some urine expressed. And so that's a good opportunity for them to taste it and make an association of this urine. Steve's urine tastes like this, basically. And so then when they go into the water and they taste a specific urine, they're like, oh, that's Steve. Steve was here a little bit ago. So this is their theory. What this sounds like to me is something that a lot of mammals do that involves the tasting or smelling of urine. For example, giraffes, they will actually taste the urine. The males will taste the urine of the female to see if she's an estrus before they attempt to mate. So they fully, they kind of do their special dance where the female goes, okay, yeah. Basically, you want me to pee for you? I'll pee for you. So she pees, and then the male tastes it and goes, yes, now is the time. She's an estrus. There's also this whole thing called Flehmen response, which is where mostly hoofstock, but also cats, there's a lot of different mammals will smell the urine of a female, and and they'll do this crazy face, which is the Flehmen response, where they curl up their front lip like completely. And it's actually to expose the specific skin above their teeth so that the pheromones can interact with the
vomeronasal organ.
Yes, also called the Jacobson's organ, which is the thing that snakes have to be able to, quote unquote, taste the air, right? It's the
That
same organ,
supposedly
which is
we
the
don't
whole-
have because we haven't found it yet.
Right. And so this is an olfactory sense organ. So they consider this a smell-based organ. This seems too similar.
Yeah,
For
so similar.
that to not be part of this. And I was not part of this research team, and I don't know if they tested, if estrus timing had anything to do with any of this, or if it was related with any mating opportunities or anything like that. But this is where I would research next if I was in charge of these dolphins, is because it really feels like it is somehow related to this opportunity. Dolphins came from land mammals. So they have actually a lot more in common with these animals that have phlegm responses on land than we like to think, because they feel so different. But so that's what's really ringing true to me about this study. The other crazy thing about this is that the researchers actually think that they might be able to use this information to have impacts on human obesity. So stick with me. They believe that the same gene that allows dolphins to identify the lipids in urine that they use to identify individuals, also identify other information from them, that these are the same that are present in humans, where it helps them to know if they've had enough to eat. It's the same lipids.
Really?
Yeah.
Huh.
So they think that studying the gene in dolphins it's the same gene, not the same lipids. It's the same gene. So they think that studying the gene in dolphins could improve understanding of how we regulate our own intake. Seems like a bit of a jump to me. But they think that it's related. So they think there's something to that. But also knowing that there's human-caused pollution, like oil spills and other chemical runoff, that also are going into the water, mean that there could be an impact to dolphins' ability to signal one another. So there's these two other crazy ideas. So one is that somehow you could use this knowledge of this gene to help with human obesity. But the other crazy idea, less crazy, but still kind of very interesting, is that you have to keep in mind when you are spewing pollution into waterways that you could be impacting dolphins' ability to taste each other's pee. So consider that.
I will definitely be considering it the next time I try to pollute a waterway.
But think about the dolphins trying to taste each other's pee. Don't pollute.
I'm gonna say I think that the link to human obesity is kind of a stretch, but I do appreciate the implications for. Here we have an animal who exists in its, we go through air, and if you walk through a plume of smoke, you walk through pollution that you can smell, or, something that is noxious, that affects the way you breathe or the way you feel, it's really going to affect you. And if you're an animal like humans, we apparently, we've got pheromones, hormones, body odor, that kind of stuff, but we don't use it the same way these other animals do, obviously. But if you're an animal like a dolphin, who uses these scents in the water, these scent tastes in the water. Yeah. We're impacting all sorts of life in the water where they, use these chemicals to survive.
Yeah. Meanwhile, survival. If you're a baby leatherback turtle, it's all about the moon.
Yes, it is.
And not how you would think, actually. So when you're a sea turtle hatchling and you emerge from your nest, it's usually at night, and you have to crawl towards the ocean. This is called sea finding. You have to do it quickly and efficiently, because otherwise you will get ate by birds, crabs, raccoons. It's a harrowing journey. And the way that you would do this as a baby turtle, I'm just going to keep talking to you, the audience, as you are all baby turtles. You would figure this out because the sea would actually be dimmer than the land, or brighter than the dimmer landward horizon. So the idea is the moon is out, the moon is reflecting on the ocean, it does not reflect off the beach and trees behind you. So it's brighter towards the ocean. So you got to walk towards the brightness. You got to shuffle towards the brightness. Sorry, I forgot turtles.
Little baby turtle shuffle.
Yes. So this helps them to find the ocean even when it's uneven and maybe they have to go up a bank to go back down. They're following the brightness. This study looks at, this is from Florida Atlantic University, and it's looking at the difference between leatherback sea turtles and loggerhead sea turtles. And leatherbacks, Dermochelys coriacea, they often will crawl around in circles trying to find the ocean. They have trouble. It delays their entry, and of course that changes their survival. The hard-shelled turtles, the loggerheads, they are more sensitive to light. They have an easier time finding their way to water. Those leatherbacks, less sensitive, harder to figure it out, spend more time going around in circles. Leatherback eyes.
This means backwards.
Yeah.
The ones with the hard shells who have a little bit more protection are like, oh, it's so easy. And the ones that are more set and sensitive are like, where?
Absolutely.
Don't eat me.
It doesn't really make sense. So the other strange thing is then when these researchers looked closer, when they first just noticed that the leatherbacks were going around in circles over and over and over, they looked at leatherback eyes. And there was not any obvious structural adaptation that might promote improved visual function under dim lights. They didn't have a larger cornea. They didn't have a larger lens. They weren't able to gather light more efficiently. And so their eyes are inferior for this task is ultimately what it comes down to. And so the only time that they were really good at getting straight to the ocean is closer to the full moon. When it was during a new moon, when it was very dark, they had a really, really hard time. They were going all over the place. And so this is, first of all, just helpful to know with turtles because they're endangered, obviously, and light pollution can be a problem because if they're trained to go towards brightness and there is artificial light somewhere, that can mess them up. This is part of the reason that sea turtle nesting beaches are so well protected and monitored, because stuff like that can really mess them up. But the reason researchers think that these leatherbacks are so poorly equipped for their journey so early in life is that they actually are better at seeing in the ocean once they get there. Yeah. So the costs persist. Basically, the reason it's beneficial to have trouble finding the ocean originally is that they are better once they get in the ocean at detecting prey, mates, and favorable habitats. Loggerheads, alternatively, are mostly in shallow coastal waters, very clear waters, easy to navigate, all that good stuff. The leatherbacks go way deep, and so they have to deal with a lot more murky waters, darker waters, that kind of stuff. But just the way their eye is structured, researchers think it's beneficial in the long run. But that does mean that their survivorship is not great.
That's very low when they're young.
It is about 1 in 1,000 to 1 in 10,000 leatherbacks will see adulthood. So it's not great.
That's awful.
Yeah.
Wow. I had no idea that was the ratio. That's
why sea turtles lay hundreds to thousands of eggs depending on the species. And it's pretty wild.
There's a question, couldn't they assist turtles with bright lights that are well placed? So on moonless nights, wouldn't it be a great idea to take big stadium lights and shine them at the ocean?
Yes, and I would not be surprised if turtle conservationists currently do that. That sounds like something they might do.
Yeah. I mean, what can we do currently? What are the things that we can do to help these poor little turtles who die at such high rates?
Well, here's the thing. Before humans stomped on the scene, stomp, probably driving some animals to extinction as they stepped, they did fine because once they got to the ocean, they thrived and their numbers were good. Climate change wasn't happening. So they had these big clutches of eggs, and it was okay that 1 in 1,000 survived. But now that sea turtles have all these other problems, climate change, they have pollution, they have poaching, they have all these other things that are happening to them. Now it's too many things. Their populations can't handle it. Yeah. And that's why we're seeing this problem. It's not that this survivorship, and it sounds really mean, but really it was part of how it was working before. You laid a giant clutch and some of them made it. But because we're now having this kind of back end pressure on adults that didn't really exist before, they were nigh indestructible except for maybe by bull sharks or tiger sharks. That now there's this extra pressure that is all of the human impact. And so it's a problem. Now we'd like more of them to make it to the ocean.
Yeah, we would. We would like many, many more of them to make it to the ocean and not eat plastic bags that they think are jellyfish.
Mm-hmm. Or get straws stuck up their nose.
Get straws up their noses?
No.
I thought that was only a problem for elementary school kids.
Oh, no.
No. Okay.
Unfortunate.
Unfortunate. Is that all you have for the Animal Corner?
Just dolphins and turtles today. Very aquatic episode.
This is a very aquatic episode. And, earlier, plummeting salamanders.
Salamanders.
From the trees to the seas and beyond. Well, I want to talk to you about corvids. Can we talk about birds? I'm going to take it a little bit. Always, yes. I'm going to take it a little bit further for a bit of bird brain conversation. So there's been a question for a very long time. Birds have very small brains. How is it possible that some of them have incredible cognitive abilities? Corvids are a family of birds, crows, ravens, jackdaws, scrub jays, magpies. They're all in this family of corvids that are capable of such amazing feats of memory and cognitive ability. One of the things that's very interesting about their abilities is that they are very, I guess, plastic, or they can switch strategies if they need to. They can adjust what they're working on, so they're not just focused on only doing things a particular way. They can learn fairly quickly, more quickly than other species when it comes to various tests that we've given them. So there's this question of how they do it. Well, a bunch of researchers said, well, let's just get down to it and look at their brains. So they compared a whole bunch of brains between chickens, pigeons, ostriches, and some crows, carrion crows, and others. So they took a whole bunch of corvids and non-corvids and looked to see what was going on, because they're like, okay, one of the hypotheses is number of neurons. And so number of neurons in different areas of the brain. The telencephalon is kind of like the big processing area of the brain in the bird brain. And so they said, okay, maybe number of neurons there. Compared across, and they said, nope, it's all pretty much the same. Oh, interesting. Crows and ostriches have very similar number of neurons in their telencephalons, which I find very interesting because ostriches are such big birds.
They're not very smart.
And yeah, comparatively, very, very fascinating here. Yeah. And so then they extended their hypothesis a bit to suggest that maybe it's not just the overall telencephalon that's important, but parts of the telencephalon in specific. So high neuron counts in what are called associative pallial areas. And these are areas that are thought to drive flexible, complex cognition. And so they comparatively, these are the associative pallial areas would be comparable to some of the higher processing cortical regions in the primate brain that allow primates to do a lot of the amazing feats of cognition that we're able to do. So they looked in these pallial areas and they're like, oh, corvids have more neurons than chickens and pigeons, which is not really a big deal, but really their very specific areas were twice as high in corvids than in all the other birds. So the corvids had these very specific, these associative pallial areas, the mesopallium, the nidopallium, the subpallium. There's all these areas in the brain that corvids had more. They had it better. So it really does kind of add to this story that researchers are trying to figure out is what makes the corvid brain so special. And it is apparently the allotment of neurons. So where are the neurons more densely packed? Where are they? Where are the resources in the brain? And where are the connections happening? And so these associative areas, it's not just the areas, it's the fact that they're associated with each other. And so they have connections kind of between them. And that's another aspect of the primate brain, which is that there are these connections and networks that allow very complex thought to occur.
I'm glad we're figuring this out because otherwise I'd just be convinced that crows are cryptids.
They're not cryptids. Nope.
For the first half of this story, I was like, so what is it then? There has to be an explanation. If not, it's magic.
Right. And
because they're just so far and away smarter than other birds.
Right. I mean, except for maybe the parrots, the Aussines, or, there are definitely social birds that are very smart, but the corvids, they are special.
They have their tool making and strategy and tool storage and communication between each other and memories and things that are special. I don't see parrots. Parrots might be able to identify objects and learn phrases and do other really cool stuff, but it's not the same.
Yeah. Yeah. It's still pretty impressive though.
Oh, it's definitely still impressive. It just feels like on a different level of organization.
One thing I thought was very interesting. So when I was studying bird brains, the area of the brain that I focused on was the hippocampus. And the hippocampus is this area that's usually it's right on the top. So you've got in the pigeon, in the rear of the brain, it lengthens out and it becomes this very broad structure that's right on the top. It starts out kind of in the middle section. In the chicken, it's very similar, but the measurements that they did, they found that the chicken has massive numbers of neurons in the hippocampus compared to other birds, which I find very interesting. And I want to understand why that is, but I'm not a graduate student anymore or a researcher. So somebody else has to do that for me. But the carrion crow, man, beautiful, beautiful, big hippocampus, very large, clear brain structures. I'm in love with the crow brain. They also did their neuron counts to try and determine which neurons were there. And anyway, long story short, yes, corvids, they have more neurons in the right places to allow more complex cognitive thought. Right. If you think that more is better, then that's the correlation that you can therefore draw.
More is better for some things, which apparently includes neurons.
Yes. Apparently neurons in this particular
sometimes case. Specific types in specific animals. Asterisk. Asterisks. Oh, and before we put you to sleep, because I really don't want to do that. I don't like putting. I really wanted you to put you to sleep. I would just get the anesthesia out, knock you out, put you down. Which we talked about on the show is very interesting. Anesthesia. It has this wave-like. It just soothes the brain, and instead of the brain having massive activity where neurons are active all over the brain at different times and having all their conversations with other neurons and blah, 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 blah. Brain's very going on its
its own thing. When anesthesia gets in there, the brain, the neurons all go woo all together. Woo, and it's a wave around the brain. Ooh, and it's like the neurons all want to do the same thing, and they go ooh all together.
That sounds very calming.
Well, you're in anesthesia, so yes, in a sense, it is. Researchers just published this week. They were trying to look at anesthesia and the effects on brain functions and on brain activity, deep anesthesia. There's a phenomenon, it's called burst suppression. And so you have this synchronized activity and then a period of silence. This is burst and suppression. And the deeper the anesthesia, the shorter the phases of the bursts. So you have burst and lots of quiet. So the deeper you're under, the less active your brain is. And so the more silence or suppression there actually is. And, different anesthetics have different burst suppression patterns, and they vary in their mechanism of action. And so these researchers were like, what is going on in here? And so they did an fMRI study, and they decided to compare humans, monkeys, and rodents in a standardized method so that it was the same all across the board to see how anesthetized brains, all the different species brains, reacted under the fMRI and whether it showed the same pattern. And what they found is that all of the species had the same kind of burst suppression activation. However, the visual cortex in all of the species wasn't following the same pattern, except for the rats. So the visual cortex of monkeys, of the primates, of humans, the visual cortex was like, I'm not going to play along with your burst suppression game. I'm going to do what I want and have my own activity the whole time. So the visual cortex is like, anesthesia, whatever. Except in rats. And they found that in rats, the visual cortex did exactly what the anesthesia wanted. It did the burst suppression. It did the pattern the same exact way. And so the question now that these researchers are raising is, what's the difference in the rat brain? Why is the visual cortex following the burst suppression pattern when in the macaques, the marmosets, the humans, the visual cortex obviously was like, one of these kids is doing its own thing. Why was it different? And if it is this different, should rats be used in brain models that are used for anesthesia, that are used for neural treatments, that are used for so many different, eventually the research always ends up in humans before it, in clinical trials before it gets used. But we find this big of a difference in the pattern of activation. And it really makes you wonder about the models that we use for our research.
What could this mean? How could this manifest physically and practically if the visual cortex isn't playing along with anesthesia? What could that result in?
I don't know. Right.
I guess
that's another big question, right? What is happening? Why is there continued activation within the visual cortex when it is part of the brain? It's not like it's completely separate, but the one thing that they ask is like, maybe it's because primates are primarily visual, that there's something to do with even just the way the visual cortex is activated during deep sleep, or the way it's connected to other areas of the brain, because it is such a primary sense for our survival, that maybe there is some kind of some aspect of its underlying importance.
Right. Yeah. Primates have binocular vision, which is the forward-facing eyes usually associated with predators.
Right.
And
Rats
so rats, of course, have the the vision on the sides. I forget what the fancy word for that is, but it's what prey has.
Yeah.
So they're inputting that information completely different because it's not a continuous field of view like binocular vision is. So they're processing the information completely different, but also if you want to think about it evolutionarily from like what that information is giving them, it's also different. If they're constantly looking for predators, but monkeys and primates are foraging, using them for social interactions, hunting, it's kind of all related.
Yeah. I don't know. But I mean, they found this. They didn't expect to find this. And they don't understand why there's a difference.
Mm-hmm.
What
Yeah,
does
see
it
what cats
mean?
do. Cats do, just pick another predator that's like similarly quote unquote uncomplex, like a mouse, right? And see what's going on.
Stop calling my cats uncomplex, you dogophile.
You know what I mean? Dogs are quote unquote uncomplex also. All those quadrupeds.
I got you.
Kind of, just stick figure mammals. Lump them all together, right? Carnivora.
The carnivora, yes. Oh, I don't know. We're just wasting our time until the fungi come and eat us all. So
yeah. That's fine. If I get to see a sentient slime mold, I will succumb to it. It'll be a great day.
Oh, you're amazing.
You can have it.
Me. That's fine.
Just take it. It's fine. You deserve it. You've earned it.
Well, that's it for me. I got brains, animal brains, corvid brains. You had the P and the C.
Yeah.
Thank you for bringing that, Blair.
Of course. I like to keep it light.
I know. I like it. We like it light. We like it fun. We like it enjoyable. That's all we want. Have we made it to the end of the show? Did we do it?
We did it.
Oh my gosh. Definitely under 90 this
once
week.
we're done with all this stuff, it'll be,
Yeah, tight 90. That's what we're heading for. Oh my goodness. Everybody, thank you so much for listening to the show this week. If you have any questions, send them to us. We might be able to answer them on the air. We like answering your questions. We could be doing that right now. If you had a question for us, we could be doing a quick answer segment if you had a question. But anyway, in the meantime, it's time for me to say thanks. Give shout outs to the people who help with the show. Fada, thank you so much for your help on show notes, show descriptions, on social media, so many things. I appreciate you so much. Identity 4, thank you for recording the show. Gord, Arnlore, others who help to keep the chat rooms happy and safe and kind places to be. Thank you for being there. Rachel, thank you for editing the show and for your assistance. And I want to say thank you very much to our Patreon sponsors. Thank you to Teresa Smith, James Schofer, Richard Badge, Kent Northcote, Rick Loveman, Pierre Velazar, Ralphie Figueroa, John Ratnaswamy, Carl Kornfeld, Karen Tozzi, Woody MS, Chris Wozniak, Dave Bunn, Vegard Chefstad, Hal Snyder, Jonathan Stiles, a.k.a. Don Stylo, John Lee, Ali Coffin, Maddie Perrin, Gaurav Sharma, Ragan, Dunn Mundus, Stephen Albaran, Daryl Myshak, Stu Pollock, Andrew Swanson, Fred S104, Sky Lee, Paul Ronovich, Kevin Reardon, Noodles Jack, Brian Carrington, Matt Bass, Sean and Nina Lamb, John McKee, Greg Riley, Marquess and Flo, Jean Tellier, Steve Leisman, a.k.a. Zima, Ken Hayes, Howard Tian, Christopher Rappin, Dana Pearson, Richard, Brendan Minish, Johnny Gridley, Remy Day, Flying Out, Christopher Dreyer, Otiam, Greg Briggs, John Atwood, Rudy Garcia, Dave Wilkinson, Rodney Lewis, Paul, Philip Shane, Kurt Larson, Craig Landon, Sue Doster, Jason Olds, Dave Neighbor, Eric Knapp, E.O., Kevin Parachan, Aaron Luthen, Steve DeBell, Bob Calder, Marjorie, Paul Disney, David Simmerly, Patrick Pecoraro, Tony Steele, and Jason Roberts. Woo-hoo. Thank you all for your support on Patreon. And if you want to support us on Patreon, hear your name at the end of the show, head over to twist.org and click on that Patreon link on next week's show.
We'll be back on Wednesday at 8 p.m. Pacific Time, broadcasting live from our YouTube and Facebook channels and from youtube.com/thisweekinscience.
Want to listen to us as a podcast? You can do that. Just look for us This Week in Science, wherever podcasts are found. And if you enjoyed the show, hey, get your friends to listen too.
For more information on anything you'd like to learn about that you heard here today, show notes and links to stories will be available on our website. That's at www.twis.org. And I don't know, maybe someday we'll send out a newsletter again. Who knows? It'll be a huge surprise when what's this in my inbox? What?
Rise from TWIS.
It'll come around sometime.
You can bug us about that newsletter directly by emailing Kirsten at thisweekinscience.com, Justin at twisminion@gmail.com, or Blair Baz at twis.org. We like to keep it complicated by not letting anyone have the same emails. Anyway, just put TWIS in the subject line so your email doesn't get spam filtered into oblivion. Wait, I didn't make up a funny thing. What would you say, Blair?
I would say be sure to put TWIS in the subject line, or your email will be collected by a crow because it'll look nice and shiny. They'll take it to their tree spot. They'll polish it and change it and morph it into a tool. They'll store it very carefully, but we will never ever see it because they're going to protect it from the scary humans.
And I'm not good at watching where the crows store their stuff. So
no. Just put TWIS in the subject line. You can also ping us on Twitter, where we are at TWIS Science, at Dr. Kiki, at JacksonFly, and at Blair's Menagerie. We love your feedback. If there's a topic you would like us to cover or address, or a suggestion for an interview, please let us know.
Oh, yeah. And we will be back here again next week. And we hope that you'll join us again for more great science news.
And if you've learned anything from the show, remember,
it's all in your head.
This Week in Science. This Week in Science. This Week in Science. This Week in Science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth, and I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in 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 Science.